Welcome students. This is for 4th semester students. Plant tissue culture in unit 3, lecture number 2. All 4th semester students listen to this class and prepare your notes. Actually, this is a very important class. About many times they have asked about questions in this media which is used, especially MS media, and the basic development of plant by tissue culture, basic techniques. They used to ask 5-mark question and sometimes 10-mark question. When they ask, this should be written very clearly in the part of after the different types of tissue cultures and basic definitions which we have discussed in the last class. So it's very important. So you have to listen to the class and prepare your own notes. So when you come to the syllabus, we have already seen the history and development of plant tissue culture in a short manner and types of culture also. Today we are going to discuss about the nutritional requirements, growth and their maintenance. It will continue little more in the next lecture, but here the nutritional requirements, what is MS medium, all these things we are discussing today. So when you want to talk about nutrient media for plant tissue cultures, one of the most commonly used media for plant tissue culture was developed by Murashik and Skoog. Murashik and Skoog, short form MS. They used to say MS media. Murashik and Skoog, they prepared for tobacco tissue culture initially. The function of the medium, first we should know function of the medium. Whatever you have the knowledge in microbiology in last semester what you studied, you can utilize here little more, but it will be easy for you once you prepare the notes. It will thorough your knowledge what you have studied before. Every semester when you come up and up, it's revising and giving some advanced knowledge for your career. So it's necessary for you to make use of the previous semester what you learned from microbiology. So the function of the medium provides first one water, and you can see it provides mineral nutritional needs. It gives mineral nutrition needs including nitrogen, phosphorus, why not potassium. These all are sulfur which is needed for the development of the plant. Provide vitamins. It provides vitamins. Provide growth regulators. Already the 4th semester students will come across all the growth regulators clearly in your cultivation collection chapter. So this is also provide growth regulators in our in vitro study like plant tissue culture. Provide growth regulators and access to atmosphere for gas exchange. Access to atmosphere for gas exchange. Removal of plant metabolite waste. So function of media, when you want to write, I have made into point by point. If the student specify, it will be very easy for you to score the mark, very easy to remember and to write in the university examination. So see here, components of MS media, Murashik, Skoog. It is MS media in short form we can remember. In MS media components, when you see number 1, inorganic salts, number 2, carbon sources, number 3, vitamins, number 4, growth regulators, number 5, organic supplements. So a student can easily remember the components in 5 categories. Organic supplements which are available, organic supplements also. So these 5 points are the main components of MS media. But we have to describe in detail. So when we come to the media of the plant and the cell culture, we can talk about number 1, inorganic salts. In that inorganic nutrients, it includes mineral salts that are important for the growth and development of the plants. It is categorized into 2 groups. One is macronutrients or nutrients like calcium, magnesium, nitrogen, and micronutrients like copper, iron, and zinc. Number 2 category, carbon sources, sugar or glucose. At 2 to 4% are suitable carbon sources which are added to the basal medium. Fructose, maltose, and other sugars also support the growth of various plant cells. So an optional concentration should be, not optional, optimal concentration should be used to establish the efficient production process of useful metabolites. Numbers, that is C, vitamins. In Murashik's cook or MS medium include myo-inositol, nicotinic acid, pyridoxine hydrochloride, thiamine hydrochloride. Along these vitamins, thiamine is an essential one for many plant cells, and other vitamins stimulate the growth of the cell in some cases. So growth regulators which you have studied, it includes auxin, cytokinin, and gibberellins. It is essential for the growth and development of tissues and organs. And number E, when you come to organic supplements, these supplements include casamino, you can say casamino acids, peptones, yeast extracts, malt extracts, and cocoa in order to stimulate the growth of the cells. Organic supplements are sometimes added to the medium nut milk. So casamino acids are also of the natural occurring amino acids and particularly rich in essential amino acids. So you can see it is available in the market, casamino acids. Casamino acids have undergone a nearly complete acid hydrolysis of the casein. We know casein is a protein which is present in the milk until all the nitrogen is converted to free amino acid, intended for laboratory and manufacturing use only. When you are coming to the in vitro study in tissue culture, plant tissue culture, we can use this cas amino acids. So coconut milk is also another organic supplement. Coconut milk is also known as a supplier of growth regulators. They have growth regulators. In order to stimulate the growth of the cells, organic supplements are sometimes added to the medium. The Morashik and Skoog MS medium, preparation of the medium, how to prepare. We know the components. Now we should know components, what all should be added, how to prepare. Components of the MS medium preparation, which we told the 5 points, was short, little more detail when we want to talk about. When you see, number 1 is inorganic salts. Number 2, you can say vitamins. Number 3 is calcium chloride. Number 4, potassium iodide. Number 5, you can say it is 2,3-dichlorophenoxyacetic acid, which is a derivative of plant hormone auxin. And number 6, you can see that N-naphthalene acetic acid. It is also another plant hormone derivative of auxin. Number 7, kinetin, cytokinin, is a derivative of cytokinin, plant hormone. These all are main components of Morashik Skoog medium, or in short form you can say MS medium. So these 7 points you have to remember and explain what all the inorganic salts, what all the vitamins, what all the calcium chloride, what all the potassium iodide. The slides which I have told two slides before was a general idea. Now we will see in detail about the chemicals which we are using during preparation. For example, MS media is prepared as follows. Nutrient stock solution should be stored in the refrigerator or freezer, not refrigerator, in freezer. And you can see inorganic, A is inorganic salts, ingredients in milligram per 100 ml. The numbers will be shown in milligram. In 100 ml you are preparing. Boric acid 620 means to 620 milligram. Manganese sulfate is 2230 milligram means 2.23 grams of manganese sulfate. Zinc sulfate 860 milligram, and sodium molybdate is 25 milligram. Copper sulfate only 2.5 milligram, and cobalt chloride is 2.5 milligram. And number 2, vitamins we have to add. They also should be stored in a freezer, and vitamins in milligram per 100 ml is given below. Nicotinic acid 100 milligram. Thiamine hydrochloride 1000 milligram, that means 1 gram. Pyridoxine hydrochloride is 100 milligram, and myoinositol is 10,000 milligram, that means 10 grams. Number 3, it is needed for you to think about calcium chloride. When a calcium chloride 15 grams for 100 ml should be prepared and to be kept in the freezer. Potassium iodide is also an important component in MS medium, stored in an amber colored bottle in refrigerator because it is light sensitive. So potassium iodide 75 milligram in 100 ml. And number 5, 2,4-dichlorophenoxyacetic acid. This is an auxin derivative of a plant hormone. Auxin plant hormone derivative. Dissolve 50 milligram of 2,4-dichlorophenoxyacetic acid. In 2 to 5 ml of ethanol, heat slightly and gradually dilute to 100 ml with water. Store in refrigerator. And number 6, 1 naphthalene or naphthalene acetic acid is also derivative of auxin plant hormone. It's a derivative of auxin plant hormone derivative. Dissolve 50 milligram of 1 naphthalene acetic acid NAA in 2 to 5 ml of ethanol. Heat slightly and gradually dilute to 100 ml with water. Store in refrigerator. Number 7, kinetin is a cytokinin. It is also hormone, cell division plant hormone. Dissolve 21.5 milligram of kinetin in a small volume of 0.5 normal HCl by heating slightly and gradually diluting to 100 ml with distilled water. Store in refrigerator. Similar procedure can be used for other cytokinins if it is needed to be added for a specific plant when you are cultivating or when you are preparing, when you want to develop by tissue culture. It varies. Addition of hormones, addition of vitamins in an MS medium can be varied or can be altered according to the requirements. Other media substances which promotes growth of the tissue culture, like protein hydrolysates. Very interesting. Protein hydrolysate is interesting. You have to remember what are they. They are important growth promoters or promote growth in the tissue culture. Soya protein hydrolysate, yeast extract, fruit like banana extracts, coconut milk fresh, or it can be pasteurized. See, this is the soya protein hydrolysate, and this is the yeast extract, and this is available from the banana fruit plant extracts, coconut milk fresh and pasteurized. This can promote growth of tissue culture. Students should remember this organic natural supplements which can promote growth in tissue culture. Phenolic compounds like fluoroglucinol, which we are using in the lab for staining the lignin cells in our section when you are doing practical. This fluoroglucinol is a phenolic compound, stimulates rooting and shooting sections and shoot sections. Activated charcoal, which we are using as an adsorbent, various places, in clinical also, and in lab also, and in chemistry. It's an adsorbent or activated charcoal is used as a detoxification agent. Detoxifies waste from the plant tissue and impurities. Adsorption quality vary. Concentration normally used in 0.3% or low. It adsorbs the secondary product secreted by the tissue culture. Charcoal for tissue culture acid wasted and neutralized, never reused. Charcoal for tissue culture, when you are using tissue culture acid wash and neutralized, never reused for this culture. Controls the supply of endogenous growth hormones. What is its action? It controls the supply of endogenous growth hormones. So interesting to know about other growth promoters used. And demineralized water. We are using demineralized water also as a media for supportive promoting growth in plant tissue culture. Solidification agent, which is also very important thing in preparation of the medium. What is this solidification agent? But you know very well, very interesting, and it is when you see the chemical names, students have already come across these points. So what are they? Number one, agar is a solidifying agent. Number two, alginate. Number three, carrageenan or carrageenan. And number four, gelatin. Number five, starch. And number six, silica gel. Number seven, hydroxyethyl cellulose. Number eight, polyacrylamide. So this 8 solidifying agent which we come across most of them in pharmaceutics in last semester you might have come across. Here we are using in tissue culture, you have to solidify and they should act as a nutrient soil in an open atmosphere. How it gives, it should become into the lab in the tissue culture in our in vitro study. So you can see very clearly agar, alginate, carrageenan or carrageenan usually pronounced, gelatin, starch, silica gel, hydroxyethyl cellulose, polyacrylamide. These 8 are solidifying agent. Solid media are often preferred because it improves oxygen supply and support culture growth. So very good point the student should remember. Why solidification? Therefore, substance with strong gelling capacity is added into the liquid media. The most common substances which we have seen above is agar, other or alginate, carrageenan, gelatin, starch, silica gel, hydroxyethyl cellulose and polyacrylamide. Agar is an extraordinary resistant to enzymatic hydrolysis at incubation temperature, and only few bacteria exist which are capable of producing degrading enzymes. So it's a protective agar. So that's why we are using agar in the medium. An iron source is needed in the form of EDTA. Ethylene diamine tetraacetate can make an valuable chelate with metal ions. So EDTA with iron can be used as an iron source. pH of the medium should be maintained or the range of 5.6 to 6 before autoclaving the culture medium. Antibiotics are also for preventing of excessive contamination in the tissue culture or culture medium. Commonly used antibiotics are carbapenem, remember 500 milligram per liter, and augmentin 250 milligram per liter. So these are the two antibiotics which we use to control the culture MS culture medium of tissue culture to prevent the excessive contamination. And a certain volume of such stock solution is mixed and an appropriate carbon source is added to the mixture. After pH is adjusted to around 5.5 with 0.2 normal potassium hydroxide or 0.2 normal hydrochloric acid diluted on deionized water, you have to use deionized water, is added to the mixture up to the certain volume required. Agar 0.6 to 1% weight by volume is added for a solid medium. Medium such prepared is distributed to the vessel such as Elmer flask. The student should know what is Elmer flask. It's like an conical flask with measuring in the, you can see the picture, made up of borosilicate. Is a German product. An example, 50 ml of medium or 300 volume of Elmer flask also can be taken. The picture shows 500 ml Elmer flask and sterilized by using an autoclave at 120 degrees centigrade for 15 minutes. The sterilization condition should be varied based on the volume of the medium and the size of the vessel. So we can use even autoclave. The picture one is shown for autoclave and Elmer Meyer flask. Equipments required, that also very important, which you come across. You have already seen all this in your microbiology lab. In our lab, in our college, everything is available. Students are requested to go and visit and see, observe. And these instruments, specifically, even in Cognosy lab, we have tissue culture separate department where you can go and see how it is maintained. What is laminar flow cabin. There are even aseptic chambers. This is an aseptic chamber which is having laminar flow. That is why it is open. When there is laminar flow cabinet, aseptic chamber is not needed. The cabinet or the cabin are commercially available in different sizes. They are placed in the laboratory according to the need, where needed. If there is a sterile room, the cabinets are not always necessary. With HEPA filters, if you have a large HEPA filters in the walls of the lab, there is no need for a chamber because the room itself where you are doing inoculation will be completely sterile area. Number 2, autoclave is an instrument requirement. Autoclaves of different sizes are commercially available. You can see that for moist heat sterilization, which we are using for plant tissue culture and microbiological studies. We used to use this size. Even room type, shelf types are also available. Oven for dry sterilization, dry heat sterilization, which we needed, we can use. It's available bigger than this also in our lab. Although autoclaves can be used for dry sterilization, an oven, hot air oven, is more useful for sterilization of scalpels, glasswares, and such as petri dish, pipettes, and other glasswares which you are using. And number 4, equipments for sterilization by filtration is very interesting part which you have seen, which you have studied in filtration part, how microbes can be filtered by microfiltration. You have to see this. That is a Buckner funnel, and which can be connected with a stainless steel lock ring where a nitrocellulose membrane, very fine, and even there is a tube, a rubber red color tube which can be connected to the vacuum, and a filter flask where the filtrate will be collected. So this is a very interesting microfiltration instrument with a vacuum pump is needed, which is not shown in the picture. The medium containing carbon sources and growth regulators are simultaneously sterilized using an autoclave, but sometimes aseptic filtration is favored to avoid decomposition of unstable chemicals may be present in them. Even though heat sterilization, they may exist, and after some time they may spoil our work. So it can be removed by this sterilization by filtration. The equipments are also commercially available in different sizes and different methods. Water distillation apparatus. This distillation which is present in our machine room, which is mainly for other distillation apparatus are available. This is a very interesting water distillation apparatus which can produce pyrogen free water. To prepare media, distilled water or deionized water is generally used, although tap water can be used particularly in large scale cultivation of plant cells in a large fermenter from an economical point of view, but usually distilled water or deionized water. Deionizing instruments which I have not shown in the picture, it is available in our lab. It is columns will be there, ion exchange chromatography. By means of that, deionized water can be prepared, which can be used for the tissue culture. Number 6, culture room. When you come to a culture room, to cultivate the plant cell, culture room under different temperature or a cabinet type incubator or essential they facilitates temperature and light intensity as well as a duration of light in the room and or in the cabinet are controlled under the optimal conditions. Number 7, shelves. In the room, the shelves are also with lights, visibility, shelves built from rigid wire mesh to allow maximum air movement and minimize shading should be used in the culture room. Shakers, many different shakers, mechanical, magnetical, ultrasonic shakers can also be used. Nowadays, carbon dioxide zone of shaking is also available. A rotary shaker or a reciprocal shaker is necessary for suspension cultures. Culture room or incubators, you can see this is a culture room incubators. An incubator, a person who is a researcher or a scientist who is opening the incubator, you can see the double door system. Cultures and incubated or cultures are incubated on shelves or in incubator under specific condition of temperature, humidity, air circulation and light. Incubation chamber or area should be both light and temperature controlled devices managed for 24 hours period. Generally, high output cool white fluorescent light is preferred for a photo period duration. Photo period duration, it's an important point. Photo period. Plant having an atmosphere for the development and the preparation of their food, photosynthesis. Duration, specific period for total darkness as well as for higher intensity light. With a temperature range of 25 plus or minus 2 degrees centigrade, range of 18 to 25 degrees centigrade can be used. The rooms are required to be maintained at a relative humidity up to 70 to 75 percentage, range of 20 to 90 percentage, controllable plus or minus 3 percentage, and uniform forced air circulation should be provided. Adequate aeration is required for cell to grow. I made this slide just to make the importance of adequate aeration. There are various methods. Apart from this also, new technologies are available. Tissues which are cultured on semi-solid medium do not require any special method for aeration, but tissues which are grown in suspension cultures require special device for aeration. Number one, you can have this automatic shakers, ultrasonic shakers, carbon dioxide atmospheric shakers. Placing the culture vessel on the liquid medium on an automatic shaker. Number two, you can see a filter paper surrounding the plant which is grown in a test tube in a plant tissue culture. Very clearly you can see in this picture. Here the two ends of the filter paper are dipped in the medium, and the middle horizontal portion on which the tissue is placed, that means explant, remains above the level of the medium. This method is called a filter paper bridge method. Interesting. Students should know filter paper bridge method. How the filter paper was folded and kept in the medium in a test tube and in a beaker. It was shown with a plant after developing also for the plant, for the student to be understood clearly. Number three, this you know very well, continuous process where a stirrer and a sample can be removed from the bottom and a filter air can be supplied, and as well as the exhaust for the air also given in the picture. So the passing sterilized air through the medium and by stirring the medium as shown in the picture by a magnetic stirrer. This is, you can see, we are using cotton and we are using adsorbent cotton covered with a cheesecloth. Culture vessels are closed with non-absorbent cotton covered in cheesecloth. So these all are for aeration, for adequate need of oxygen or adequate need of the aeration for the development of the plant. So proper aeration prevents the entry of microorganisms. If you have this type of control filters and using adequate aeration for the cell growth. Basic method required or a technique, basic methodology, techniques of tissue culture. So what are the basic methods, which are the points which you should mention? Number one, preparation of a suitable nutrient medium, which we have just now discussed. As per the section of the plant medium is autoclave after sterilization. What is its size? What is its quantity? What type of culture you are going to prepare? These all decides the nutrient medium. And number two, selection of the explant. First prepare the medium and now you are selecting the explant. An excised part of the healthy plant or a health plant to be used, such as bud, leaf, root, seed, etc., can be selected. When we come to sterilization of explant, when you want to sterilize, you have to remember these chemicals by sodium hypochlorite, mercury chloride. I have made it somewhat visible by means of coloring it. Sodium hypochlorite, mercury chloride. And there are many, at least you can remember these two. And wash aseptically by 6 to 10 times with sterilized water. Then comes inoculation, transfer. This sterile explant is inoculated on a solidified nutrient medium under aseptic conditions. And number 5, incubation. Cultures are incubated at of 25 plus or minus 2 degrees centigrade and at a relative humidity up to 50 to 70 percentage for 16 hours of photo period, which I told, photo period, the environment for the plant, for the development of its tissues and development of the plant needed for the various chemical changes, importantly photosynthesis. So the photo period will be given by the incubation in a proper temperature. The 6th one is regeneration. The plantlets regenerate after transferring a portion of callus culture, callus which is developed in another medium, and induction of roots and shoots or directly from explants. That is regeneration. Number 7, hardening is the gradual exposure of plantlets for acclimatization. Acclimatization means the environmental conditions for a plant to develop normally in outside. But here also you need to give an acclimatization to the environment condition. That is called hardening. So an interesting word which the student should know. What is acclimatization in hardening? And number 8, plantlet transfer. After development of the plant, it is transferred to the greenhouse or the field condition in the open air according to the requirement for the development of the plant. So this is a picture which gives you about what we have discussed in this class. Starting from a plant, it's a cycle actually. You are preparing one explant from this plant, washing it, surface sterilization with what? Sodium hypochlorite. Even mercury compound can be also used. And trimming in sterile medium, MS medium which you have prepared, and several washes with distilled water should be done, and you have to inoculate in the medium. And you can see that in MS medium, the explant of a stem is an example. It can be a root, it can be a bud, it can be any part. Inoculation finally trimmed and established in the medium. That is what here it is shown. And now you can see the development of callus from the explant when you are incubating at 25 plus or minus 2 degrees centigrade with a humidity of 50 to 70 percentage of relative humidity for 16 hours, and then it is converted, it is made into subculture by shakers, and you are preparing even subculturing. You can have single cell protoplast culture can be prepared from a callus. So now you can see the development of the bud, and you can see the micro shoots development and development of root from the plantlet, and then it is transferred to the greenhouse or to the environment open ground according to the selection of the soil. It is in a polythene bag, it will be covered, and then it will be transferred to the open. Then you can see the development of the plant in the field as shown in the picture. So it's an interesting cycle which a student can easily remember, draw the diagram and make it clear what we have discussed. And this is also another one picture to see the original picture, how that one is schematic diagram. You can see selection of the plant, any part, and then preparation of the explant aseptically, inoculation. After inoculation, you can see in the test tube. This is various stages of the plant which is growing in the lab, and the plant is already transferred to the greenhouse and hardening stage. Regeneration of the plant from an explant. This is a picture from the lab. And cultivation of the plant tissue culture, you should remember about organogenesis. Sometimes they will ask in a 2-mark question, what is an organogenesis? Organogenesis, you can see all organs, maybe root, leaf, stem or a flower or a bud, you are getting a whole plant. So organogenesis is a process involving redifferentiation of meristematic cells present in callus into shoot, bud or root or even whole plantlets. The formation of organ is called organogenesis. So then what do you mean by embryogenesis? Embryo, and it develop a whole plant. What is the difference? Embryo is defined as the earliest recognizable multicellular stage of an individual that occurred before the development of characteristic organic or organs of the given species. We know that from the seed or seedlings or embryo. Production of embryo like structure from a callus, cell culture, and a proliferate development from an explant is known as embryogenesis. From the callus, you develop an embryo. That is called embryogenesis. In higher plants such as embryos, usually rise from zygote formation and are termed as zygote embryos. Advantages of organogenesis and embryogenesis: the efficiency of the process, reduction of labor cost and time, the formation of plantlets in fewer steps, the potential of production of much higher number of plantlets, and morphological and cytological uniformity of the plantlets. Production of several plants on commercial scale, including food crops, vegetables, species, and fruits, medicinal and aromatic plants. So this was the today's class, which I want to tell about the student again. Kindly listen to my class, prepare your notes and submit to my office. It's an interesting for you to give, and most of the students are obliging and very disciplined. Thank you. We will see in the next class.